The Cell Theory by Rhiannon and Karina.
Ideas about the scientific basis of life have changed a lot throughout the years. The discovery of the cell was a breakthrough that shed light on how healthy organisms function, and what are the causes that make them unhealthy. Today, the scientists are able to understand how the cell continues to grow. The spread of information about cells was through researchers collecting evidence about structures and functions of living things. They have communicated their ideas and findings to other scientists who then repeat their experiments and add their own ideas. As the information becomes more known to scientists, they have expanded and revised their explanations. The inventions of new technologies help scientists to take away their limits of understanding the cell. The process helped us to understand our current understanding of the cellular basis of life.
Robert Hooke looked at a thin slice of cork from the bark of an oak tree using a crude compound light microscope. The magnification for this microscope was 30 times. When looking at the cork with this magnification, it seemed as if it was made of hundreds of empty boxes or tiny rooms. These rooms reminded him of monks' rooms in the monastery, so he decided to call them cells. At the time, he had no idea what he was looking at was actually a wall of a dead cell. But Hooke never realized the significance of his discovery because it wouldn't be till decades later that it would be confirmed that the cells were common in all organisms. After reading his book, Graphia, which included pictures and detailed descriptions of his discoveries, other scientists were encouraged to further his research by looking for cells in other organisms.
In 1674, a Dutch liniment merchant named Anton van Leeuwenhoek became the first person to describe a single-celled microorganism. He studied a variety of things including blood cells, pond water, and leftover from his teeth. Van Leeuwenhoek made magnifying glasses, so when he looked through the lenses he saw tiny particles that looked like animals. He then named them animalcules, and these were the first observations of single-celled organisms.
Schleiden and Schwann thought that from crystallization of non-living materials, or by budding from the surface, cells would form from other cells. German scientist Rudolf Virchow corrected these misconceptions by doing experiments that showed the bone cells could develop from cartilage cells. Also, he observed cells dividing from multicellular organisms. His conclusion was that the cells divided to produce more cells. Where a cell exists, there must have been a pre-existing cell.
And that leads us to the cell theory. The cell theory has been replicated and reformed a lot throughout history. These conclusions form the basis of an important theory called the cell theory, and it states all organisms are made up from one or more cells. Cell is the smallest functional unit of life. Cells are produced from other cells. There are important influences among plant cells, animal cells, and single-celled organisms. Even so, all cells share several basic similarities, which leads us to our descriptions about each cell. Cell wall. The cell wall is a thick, rigid membrane that surrounds a plant cell. The layer of cellulose fiber gives the cell most of its support and structure. The cell wall also bonds with other cell walls from the structure of the plant. Vacuole. A large membrane-bound space within a plant cell that is filled with fluid. Most plant cells have a single vacuole that takes up much of the cell. It helps to maintain the shape of the cell. Chloroplast is an elongated or disc-shaped organelle containing chlorophyll. Photosynthesis takes place in the chloroplast. Cytoplasm. The jelly-like material outside of the cell nucleus in which the organelles are located. Heterogeron. Spherical to rod-shaped organelles with double membrane. The inner membrane is folded many times, forming a series of protections. The mitochondrion converts the energy stored in glucose to ATP for the cell. Golgi apparatus is a flattened, layered, sac-like organelle that looks like a stack of pancakes and is located near the nucleus. The Golgi body packages proteins and carbohydrates into membrane-bound vesicles for export from the cell. Ribosome, a small organelle composed of RNA-rich cytoplasmic granules that are sites of protein synthesis. Endocytoplasmic reticulum, a vast system of interconnected, membranous, folded, and convoluted sacs that are located in the cell's cytoplasm. Rough ER is covered with ribosomes that give it its rough appearance. The rough ER transports materials through the cell and produces proteins in sacs called cytosine, which are sent to the Golgi body or instead to the cell membrane. ER is a vast system of interconnected, membranous, folded, and convoluted tubes that are located in the cell's cytoplasm. The space with the ER is called the ER lumen. Smooth ER transports materials through the cell. It contains enzymes and produces and digests lipids and membrane proteins. Smooth ER buds off of rough ER, moving the newly made proteins and lipids to the Golgi body. Nucleus, a spherical body containing many organelles including the nucleus. The nucleus controls many of the functions in the cell and contains DNA. The nucleus is surrounded by the nuclear membrane. The only difference between an animal cell and a plant cell is an animal cell doesn't have a cell wall to repel water off of and doesn't have chloroplast because an animal cell doesn't need to go through photosynthesis.